For the next part of our evening, we're honored to have Dr. Peter Marks as our guest. Dr. Marks is director of the FDA Center for Biologics Evaluation and Research, commonly known as CBER. This is the area of the FDA which regulates such treatments as gene and cell therapies, protein therapies, and also vaccines. Dr. Marks received MD and PhD degrees from New York University and did his medical residency training at Brigham and Women's Hospital in Boston. Since then, he has held positions in both academia and pharma industry before joining the FDA in 2012. And he's been the director of CBER since 2016. So please welcome Dr. Marks. So to start off, a lot of the treatments under development for limb-girdle muscular dystrophies are gene therapies, particularly gene transfer therapies. And I wonder if you could sort of go into some of the comparison between the drugs like these versus more traditional drugs from both a development and a regulatory perspective.
Yeah, so thanks. First of all, thank you very much for having me here tonight. And I have to say, you're truly an amazing group of people, so thank you. I think some of what we've come to learn over the past couple of years as we've gone from having no gene therapies approved prior to 2017 to having now a total of 15 products on the market and a bunch that are almost there as well. Small molecules they generally have been developed over time. They cost a fair amount of money to develop. They have toxicities that are sometimes hard to know up front. And so it can be very challenging to develop small molecules for diseases. The good news when you get them developed is they're usually not incredibly expensive, but that's not always the case. Gene therapies have going for them the fact that if you know what gene's missing and you know what needs to be put back, there should be a rational way to make them. So that's the key thing here is that right now for many of the gene therapies that we see, they are essentially a backbone. The best thing is the analogy is like a razor handle to which the gene of interest is like a razor blade that's put into it. And we're increasingly realizing that there are some fundamental differences here that make these gene therapies really a little different than other products that we deal with, like the small molecules, because the toxicities are shared essentially usually with the razor handle between the different products. Not invariably, but it's a pretty good bet. And that may mean that we can use some tricks to hopefully move forward more rapidly and have more gene therapies approved in a very rapid manner. And probably we can discuss as we go along some of the ways that we think that we'll be able to make more haste through getting gene therapies across the finish line.
Okay. Now, as a follow-up, I have what little I know about gene therapy manufacturing is that that's also a big challenge both to developers and FDA.
Right. So gene therapies, the gene therapy vectors that are made today, they're either viruses like adeno-associated viruses. For cell-based gene therapies, they're lentiviral vectors. And now for the genome editing products, they're going to be very interesting mixtures probably of potentially of DNA and DNA RNA mixtures or RNA and proteins packaged in liposomes, essentially a delivery vehicle. This is going to require a lot of work to be able to make these in a facile way, because right now they're made very much in a boutique manner. It's not like you can go to a machine and crank these out. They're often made essentially in academic laboratories or in manufacturing facilities where there's a lot of hands-on work. And that's led to very high costs. In many of the gene therapy trials, it costs, just the cost of the product itself is $100,000 to $250,000 per dose to make the product. And so one of the things that we're very interested in doing at FDA is working with the community of manufacturers, nonprofit organizations, and we're working in a partnership with the Foundation for NIH called the Bespoke Gene Therapy Consortium, trying to figure out ways to semi-automate the production of gene therapy to try to bring down the cost and speed up the production. Because right now, some of the ability to get gene therapies into trials is not just a matter of regulators being slow. It's the fact that people can't get these made at contract manufacturers fast enough to get into people. So hopefully we'll overcome this in the coming years. I think people are finally realizing that manufacturing is the challenge that it is. This is an interesting thing. It shows you how mistakes can be made. About 5 or 10 years ago, when we were first starting to see gene therapies increase in number, a lot of people said, oh, don't worry too much about manufacturing. It'll all take care of itself. It'll be just like monoclonal antibodies. And that was a real mistake. And when you think back to it, the people who were thinking about the business models should have realized, and we should have realized, that they're very different. Because gene therapies actually, if they do their job correctly, they're one and done. Whereas these monoclonal antibodies are continue to be produced. So in some ways, the challenge of one and done creates an issue here because it's almost so good.
So LGMDs are considered rare or even ultra-rare diseases. And what challenges would you say that this creates for drug developers and for regulators?
Right. So I think there are a couple different things, and at the risk of kind of going through more than you'd like to hear about, but we have dinner, so we can go on. And like sometimes I say, oh, we'll order a pizza, and I'll tell you about it. So there are a couple different problems. One is when you have rare diseases, it's hard to, especially ultra-rare diseases, it's hard to figure out what endpoints we're going to use to get things over the finish line, because ultimately we have to show that something is showing some type of benefit in order to say something that's going to get an approval. There actually is an approval standard that basically says we have to be convinced that the products got efficacy of some type and that it's safe. And the good news is that in that regard, to say that something has efficacy, and this was mentioned already before, Congress has given us some leeway. We have the leeway of being able to use something called accelerated approval, where rather than using a clinical endpoint of how someone feels, function, or survive, we can actually use either a biomarker, which is a laboratory test, or an intermediate endpoint, which is perhaps not a final clinical endpoint. And for gene therapies, they're very well suited to using accelerated approval. And I think especially when you think about LGMD, you're talking about something that may be very well suited, because you can't wait 5, 10, 15 years for a typical clinical endpoint. What you'd like to do is look at something that you can measure in a year's time to say that something right happened when you gave your gene therapy. And that means that it may just simply be looking at whether a year out you have good protein expression, that we know then that that protein expression in some model system or in individuals who have different levels of expression, which we know happens in the community, that level is either associated with either normal or only mild problems. And so if you can show that you can match up something like that, we can use our accelerated approval pathway, and that will hopefully get us over the finish line. And then the final thing that I'll say about rare, ultra-rare diseases is something that we have as regulators as a problem globally, which is that our goal, although we are the United States Food and Drug Administration, it's not lost on us that gene therapies are probably best, they're the best hope for patients with rare diseases, not just in our country, but globally. And so the hope would be that we can help get to better regulatory harmonization around the globe so that when we have something that we feel comfortable has come across the finish line in the United States, it's easier for it to get across the finish line in other regulatory environments in Europe or in Asia, and vice versa, because that will actually, it may sound trivial, right? But expanding markets like that help actually get developers to be more excited about going into rare diseases. And though I tend to be an idealist, so I would say, look, the developers should be doing this because it's the right thing for public health. But at the end of the day, we have to be realistic. The way we will help make sure as many people in this room get cures potentially is by having a robust commercial enterprise that makes people want to go into this area. And if we can help open up markets around the globe, it's an incredible win-win. Patients get access, that's what I care about, and the companies have a larger market for their products.
That was good.
Thank you. You largely have answered like the next two questions that Kelly and I have to ask you. But so I wanted to go back to, you gave a keynote presentation at the Muscular Dystrophy Association conference earlier this year, and you actually spoke about some of these issues, having to do with making, using accelerated approval for gene therapy. I guess one question I have is, because LGMDs are ultra-rare diseases, and unless you just say, well, they're missing this protein, if we put the protein back, that's the surrogate measure. A lot of times there aren't well-established, biomarkers or surrogates. The thing is, we've never been able to treat these diseases before, so, how do we know what actually correlates with improvement?
Yeah, so I think this is where we feel like there's, I think you use whatever tools you can have at your disposal, and I think it's a combination. In some cases, it may be a combination of what we can get out of animal models and what we can get out of natural human experiments. So I'm a hematologist oncologist by training, and in hematology, there's a very analogous situation where we can measure certain genetic diseases have varying levels in different patients with the disease. And some of them, the varying level is just so modestly off that they're basically okay. In an occasional, they may have an inconvenience occasionally, but if we could get to those levels for everyone, we would know we would be okay. They don't, for instance, in a bleeding disorder, they will only bleed in a very certain circumstance, and we can deal with that. And so by getting to that, if we can take people who are at levels that bleed spontaneously and get them to that level, we can we can know that just by changing the level, measuring the level of the protein expression of the gene therapy, that's good enough. And I think the same thing, we try to use all the levers we can here. Some might be, there'll be animal models, some will be the human, and some will be complementary data, and we try to find a way to put together what can convince us that there's benefit. It's really important that there be benefit, because I think we don't want people to have, the most terrible thing that we can do as doctors is give people a medicine that doesn't actually work, right? It's false hope. And it takes away opportunity for products that actually might actually work. So I think as long as we can convince ourselves, and I think that's the exciting thing about working in this area, is to actually leverage all the science that we can. I think we'll hopefully be able to get there somewhat faster. It's going to mean taking some risks that we might be wrong. But I think what we heard from this panel here is that if there's a certain risk of that, as long as it's not a wildly crazy risk, as long as it's a very calculated small risk that we're not right, people probably will understand that we need to try what we can try to move things forward. And it's going to be uncertain, because for some of these things, I think it was beautifully said, right? If you give a gene therapy to somebody who's had a disease for 40 years, it may not, it's not going to, it's right, it's not likely. I mean, we'd love it if it could happen. Believe me, I'd love it if it could, but it's not likely to make them be able to get up and walk. But this idea of being able to stabilize things is a very valid endpoint. The problem is those are things that can take a while to show, and it's okay, as long as we can convince ourselves with a marker that it's likely that when we get the data in 5 or 10 years that we're going to get there. I don't have any problem with having things sit out there with accelerated approvals for 5 or 10 years. We could have a whole, I go to, in other more common diseases, we go to debates about whether accelerated approval is a good or bad thing. And I basically say, just stop it, because in certain diseases, the accelerated approval is going to be the only way we get there.
And you did mention a bit about the safety. And what is the FDA looking for in terms of safety and efficacy of a treatment in order to approve it? For example, how does the needed level of confidence depend on the disease, the treatment modality, and the patient preferences?
Right. So, I mean, this is really an important thing that has, I'll just back up and say something. We were talking before this about, earlier on today, a little bit about what people who work at FDA are like and how things have changed over the years. 2 decades ago, a lot of what we did at FDA, or 3 decades ago, were things for large populations where there was a lot of risk aversity, and probably justifiably so, because for people who are going to take a medicine for high cholesterol or high blood pressure or who need a blood thinner, we give them to so many people that if they had a terrible adverse effect, that's not a good thing, right? On the other hand, for a rare disease community, the risk-benefit calculation is very different, because when the outcome is virtually certain that it's going to be bad of the disease itself, people are willing to take a different risk of a therapy. Now, we can't be wildly crazy and give unsafe therapies. On the other hand, if we can define the risk so that people can make an informed decision, I think we know that there are probably, for instance, with some of the upcoming gene therapies, there's probably going to be very small risks, calculated risks, that they could cause at some point a cancer in the future for some of them that are in development. But the chance that they're going to bring benefit without that is much, much, much higher. So as long as people can understand what's the potential benefit and understand the risks, I think that risk-benefit calculus can be understood. And I have a lot of experience with that, because as a doctor who used to treat leukemia, it was a constant conversation that we would have. There was if we don't do anything, we know what's going to happen, right? Very bad outcome. if we do something, there's a small risk of a bad outcome, but a reasonably good chance that we could have a very good outcome of cure. So I think patients are in the best, and their families are in their best position to help make that decision. And our job is just to make sure that when we put therapies forth, they have at least, they have to have a certain bar. They have to meet a certain bar so that when that discussion comes up, it's the risk benefit is at least in a proper place. And what does that mean? It can't be 50-50. You know what I mean? That's not good enough. Oh, 50% chance it'll help you, 50% chance it'll hurt you. On the other hand, if it's 99-1 or 98-1 or something like this, we can live with those. And I think it's because we're just transparent about what the risks and benefit are. And I think people might, they might decide at different stages of their disease, this is worth it or this is not worth it, knowing what the alternatives are.
So how does the fact that, as it stands now, gene therapy is a one-time treatment, how does that affect these decisions of risk and benefit?
Yeah, so, I mean, one of the things about our current gene therapies is that we'd like to, I mean, this is a challenge because with AAV, the adeno-associated virus, one of the common vectors being used, once you give it once, people currently make antibodies, and it's very challenging to give it a second time. People are working on dealing with this. The next generation of gene therapies coming out that may be based on these lipid particles, they may be a little easier to give over again. But this, I mean, the hope here with these is that we get it right because we like one and done to be the case. But I think whether they cure or whether they provide long-term benefit, it's still, I think at this point in time, they're both acceptable endpoints because I think, again, whether it's cured or whether it's 5 or 10 years of benefit, for somebody who's losing function, if we could stabilize function for 5 or 10 years, that's probably an okay endpoint.
Don't let the perfect be the enemy of the good.
That is exactly, I think that's exactly right. We can't let perfect, and we tell this to our reviewers, don't let perfect be the enemy of good, because we tend, again, regulators are risk averse. It's just the nature of things. We are risk averse people. You should see my financial portfolio. It's completely risk averse. And Uncle Sam benefits from it because it's all in Uncle Sam. But we are a risk-averse bunch, but nonetheless, if we're not willing to take some risk here, we're not going to help people. And so that's, I think, the impetus to bring together a group of people where we are changing the culture at FDA from people who are highly risk-averse to people who are willing to take some risks, knowing that they could occasionally be wrong, because that's the only way they will help benefit public health. And so I think one of the great things about, just to come back full circle, patient-focused drug development meetings, the patient voice, I think probably the biggest help it gives us, rather than changing the bar of where we're going to actually approve things, is actually in helping our reviewers to actually understand what it's like to live with these diseases, what it means, what it would mean to have a therapy, so that they can feel the urgency that really needs to be there, and that makes a huge difference. So for those patients who have met with our reviewers and who have the opportunity to do so in the future, thank you for doing so, because it really helps them.
So one thing that's of great interest to the patient population is, when a drug is approved, will I fall under the approval criteria, and will before it's approved, will I meet the criteria for the clinical trials? So I wondered if you could comment a little on sort of, how. What determines the broadness of the approval or of the clinical trial and enrollment?
Right. So this issue of how drug development and gene therapy development is a little challenging in that regard because at FDA, we can give advice to companies, but it's in general, they decide who they're going to put in clinical trials. That said, I think we are taking an approach in gene therapy now that's very similar to one that we have in cancer medicine, which is to say, look, try to make the entry criteria for clinical trials as broad as it can be. And even if you have to enroll some people who might not be able to be part of the primary analysis population, at least find a way to be able to treat more individuals to enlarge the safety data set, particularly in ultra-rare diseases. And ultimately, I think the reason why we like accelerated approval endpoints is that hopefully we can then, when we give an approval under accelerated approval, give it for the widest population that we can, knowing that the hope would be that if we see activity in one part of the disease, that's going to translate into some type of activity in another part. In other words, if we see babies that don't come down with loss of function, it's probably then, and we see others who don't have the decline in function, it would be hopeful then that we can at least stabilize older people. So that's, I think, where you'll see us go. Again, will probably mean that people will be in long-term follow-up studies. It probably means that we'll be using real-world evidence to follow people over time. But that's just the nature of, I think, what we're going to have to accept for gene therapies for diseases that have a very slow natural progression, because otherwise, if we try to do, we know that the ecosystem, venture capitalists have an even shorter attention span than I do, and that's bad. And so they want the answer very quickly, and so we have to help them get products across the finish line, and then that's okay. If we can get them over the finish line in a year or two in a clinical trial, then we can follow people for years and document clinical benefit. That's okay. And I think some of these things will become obvious with time anyway.
That's great. That's so good to hear. And we heard from so many people that are excited to, we have a group of people that are willing to participate in natural history studies and obviously clinical trials too. And to say that we can try to encourage industry to broaden the patients that can get enrolled. One of the biggest things we hear is, yes, we all have hope, but there are also people who are losing hope. And I've heard our patient community members say, well, it's probably too late for me, but hopefully, we might see a cure happen in the next generation. And that's so heartbreaking to hear. So I'm so glad to hear you say that you're encouraged.
And that just takes us back full circle to this issue of manufacturing, because if some of the manufacturers, they'll say, well, we don't want to enroll, we can't afford to enroll more people. And if we could get the manufacturing costs down, hopefully they'd be able to enroll more people up front, increase. It would be good for us because we'd have larger safety data sets, and really good for patients because I think, again, there are some people who, again, if you're 50 years old with a disease, you might want to take your chance now rather than waiting 5 years for a better vector or a better gene therapy.
I think for LGMDs in particular, part of what will help will be having better outcome measures for non-ambulatory patients.
That's right. And I think that's something that the medical community will work towards. And I think we're very open and willing to work towards this. That's one of the things that I think Congress has essentially instructed us to do, to try to work with patient groups, advocacy groups, et cetera, to try to come up with better outcome measures in a variety of rare diseases.
Yeah, that's good. Thank you. I think that's
unless you have any final comments, I think we're out of questions.
So that's very sweet. I just want to say thank you all. I think we don't have all the answers. I think we're still early on in the days of gene therapy, but I think we are starting to see us come around a corner where I think we'll see the pace of progress really accelerate as we realize these therapies aren't small molecule or even protein therapies. These are kind of a new platform for hopefully correcting things that we can move more quickly through than we have in the past. So hopefully in the not too distant future, we'll see more progress in this area. But thank you so much, and thank you for all you do.
Thank you.
And thank you.